Today we'll look at the summary points of Class 12, Chapter 2, Human Reproduction. So, in this video we'll be looking the summary points of this chapter for entrance examinations like Neet. So let's get started. Introduction. Humans are sexually reproducing and viviparous. The reproductive events in humans include formation of gametes, that is gametogenesis, sperms in males and ovum in females, transfer of sperm into female genital tract, that is called insemination, and fusion of male and female gametes, that is fertilization, leading to formation of zygote. This is followed by formation and development of blastocyst and its attachment to the uterine wall, that is implantation, embryonic development, that is gestation, and delivery of baby, parturition. These reproductive events occur after puberty. There are remarkable differences between the reproductive events in male and in the female. For example, sperm's formation continues even in old men, but formation of ovum ceases in women around the age of 50 years. Topic 2. The male reproductive system. The male reproductive system is located in the pelvis region. It includes a pair of testes along with accessory ducts, glands, and the external genitalia. 1. Testes. The testes of man and rabbit are situated outside the abdominal cavity within a pouch called a scrotum or scrotal sac. The scrotum helps in maintaining the low temperature of the testes, that is 2 to 2.5 degree lower than the normal internal body temperature necessary for spermatogenesis. In adults, each testis is oval in shape with a length of about 4 to 5 centimeters and width of about 2 to 3 centimeters. The testis is covered by a dense covering called as Sturica albuginea. Each testis has 250 compartments called as testicular lobules. Each lobule consists 1 to 3 highly coiled seminiferous tubules in which sperms are produced. Let's look at a chart, that is reproductive system. There are primary sex organ, that is testis in male and ovary in female. Accessory ducts in male are rete testis, vasa efferentia, epididymis, and vas deferens. In females, the accessory ducts are oviduct, uterus, and vagina. External genitalia in male is penis, and in female there is mons pubis, labia majora, labia minora, hymen, and clitoris are the parts. Now, accessory glands are seminal vesicle, prostate gland, and bulbourethral gland in males. Each seminiferous tubule is lined on its inside by two types of cells called as male germ cells or spermatogonia, as well as Sertoli cells. The male germ cells undergo meiotic division, finally leading to sperm formation. Sertoli cells provide nutrition to the germ cell. The regions outside the seminiferous tubule, called as the interstitial spaces, contain small blood vessels and interstitial cells or Leydig cells. Leydig cells synthesize and secrete testicular hormones called as androgen. Other immunologically competent cells are also present. 2. Accessory ducts. The male sex accessory ducts include rete testis, vasa efferentia, and epididymis, also vas deferens. The seminiferous tubules of the testis open into vasa efferentia through rete testis. The vasa efferentia leave the testis and open into epididymis located along the posterior surface of each testis. The epididymis leads to vas deferens that ascends to the abdomen, that loops over the urinary bladder. It receives a duct from seminal vesicle and opens into urethra as the ejaculatory duct. The correct pathway of transport of sperms is as follows. Retestis to efferent ductules to epididymis to vas deferens. These ducts store and transport the sperms from the testis to the outside through urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening called as urethral meatus. Urethra is the shared terminal duct of reproductive and urinary system in the human male. That is, it is a urinogenital duct. The correct sequence for transport of sperm cells in the male reproductive system is as follows. Seminiferous tubule to retestis to vasa efferentia to epididymis to vas deferens to ejaculatory duct to urethra to urethral meatus. 3. Penis. The penis is the male external genitalia. Penis is made up of special tissues that help in erection of the penis to facilitate insemination. The enlarged end of penis, called the glans penis, is covered by a loose fold of skin called as foreskin or prepuce. 4. Accessory reproductive glands. The male accessory glands include paired seminal vesicle, a prostate, and a paired bulbourethral glands. The secretions of bulbourethral glands also help in the lubrication of the penis. Secretions of these glands constitute the seminal plasma, which is rich in fructose, calcium, and certain enzymes. 3. The female reproductive system. The female reproductive system is located in the pelvic region. Female reproductive system consists of a pair of ovaries along with a pair of oviducts, uterus, cervix, vagina, and the external genitalia. These parts of the system, along with a pair of mammary glands, are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth, and childcare. Ovaries are the primary female sex organs that produce the female gamete, that is ovum, and several steroid hormones, that is ovarian hormones, named as estrogen and progesterone. The ovaries are located one on each side of the lower abdomen. Each ovary is about 2 to 4 cm in length and it's connected to the pelvic wall and the uterus by ligaments. The stroma is divided into two zones, a peripheral cortex and inner medulla. The female accessory ducts. The oviducts, that is fallopian tubes, uterus, and the vagina constitute the female accessory ducts. 1. Fallopian tube or oviduct. Each fallopian tube is about 10 to 12 centimeters long and extends from periphery of each ovary to the uterus. The part closer to the ovary is the funnel-shaped infundibulum. The part closer to the ovary is infundibulum. The edge of the infundibulum possesses finger-like projection called as fimbriae, which help in collection of ovum after the ovulation. The infundibulum leads to a wider part of the oviduct called as ampulla. The last part of the oviduct, isthmus, has a narrow lumen and it joins the uterus. Uterus. The uterus is single and is also called as womb. The shape of the uterus is like an inverted pear. It is supported by ligaments attached to the pelvic wall. The wall of the uterus has 3 layers of tissue. The external thin membrane is perimetrium, middle thick layer of smooth muscles myometrium, and inner glandular layer called as endometrium that lines the uterine cavity. The endometrium undergoes cyclical changes during menstrual cycle, while the myometrium exhibits strong contraction during the delivery of the baby. The uterus opens into vagina through a narrow cervix. The cavity of the cervix is called a cervical canal, which along with vagina forms the birth canal. Hysterectomy is the surgical removal of uterus. Hysterectomy is the surgical removal of uterus. Next is female external genitalia or vulva. The female external genitalia include mons pubis, labia majora, labia minora, hymen, as well as clitoris. Mons pubis is a cushion of fatty tissue covered by skin and pubic hair. Labia majora are fleshy folds of tissue which extend down from mons pubis and surrounded by vaginal opening. Labia minora are paired folds of tissue under the labia majora. The opening of the vagina is often covered partially by a membrane called as hymen. The clitoris is a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening. The clitoris is a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening. The hymen is often torn during the first coitus or intercourse. However, it can also be broken by sudden fall or jolt, insertion of vaginal tampon, active participation in some sports like horseback riding, cycling, etc. In some women, the hymen persists even after coitus. In fact, the presence or absence of hymen is not a reliable indicator of virginity or sexual experience. Next topic is breast. A functional mammary gland is characteristic of all female mammals. The mammary gland are paired structures of breasts that contain glandular tissue and variable amount of fat. The glandular tissue of each breast is divided into 15 to 20 mammary lobes containing cluster of cells called alveoli. The cluster of alveoli secrete milk which is stored in the cavities or lumens of alveoli. The alveoli opens into mammary tubules. The tubules of each lobe join to form mammary duct. Several mammary ducts form a wider mammary ampulla which is connected to lactiferous ducts through which milk is sucked out through nipple. Gametogenesis. The primary sex organs, the testes in males and the ovaries in females, produces gametes, that is, sperms and ovum respectively, by the process of gametogenesis. Spermatogenesis. In testes, the immature male germ cells, that is, spermatogonia, produce sperms by spermatogenesis that begins at puberty. The spermatogonia, singular spermatogonium, present on the inner wall of seminiferous tubules, multiply by mitotic division and increase in numbers. Each spermatogonium is diploid and contains 46 chromosomes. Spermatogonia, after mitosis and differentiation, becomes primary spermatocytes which is diploid, and after meiosis 1 it gets arrested, it don't get arrested like in females. So after meiosis 1 we get secondary spermatocytes which is haploid. Then after meiosis 2, which won't get arrested as in females, it becomes spermatids, that is also haploid. Then after spermiogenesis or differentiation again, it forms spermatozoa which is also haploid, and after spermiation it gets released from the seminiferous tubules. Some of the spermatogonia called as primary spermatocytes periodically undergo meiosis. Spermatogonia cells called as primary spermatocytes periodically undergo meiosis. A primary spermatocyte completes the first meiotic division, that is reduction division, leading to formation of two equal haploid cells called as secondary spermatocytes, which have only haploid set of chromosomes. The secondary spermatocytes undergo second meiotic division to produce four equal haploid spermatids. Spermatids are transformed into spermatozoa by process of spermiogenesis. After spermiogenesis, sperm heads become embedded in the Sertoli cells and are finally released from the seminiferous tubule by the process called as spermiation. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone, that is GnRH. This is a hypothalamic hormone. The increased levels of GnRH then acts at the anterior pituitary gland and stimulates secretion of two gonadotropins. One is luteinizing hormone LH and follicle stimulating hormone, that is FSH. LH acts at the Leydig cells and stimulates the synthesis of secretion of androgens, which is male hormones. Androgens in turn stimulate the process of spermatogenesis. FSH acts on Sertoli cells and stimulates the secretion of some factors which help in the process of spermiogenesis. Next topic is sperm. Sperm is a microscopic structure composed of a head, neck, and a middle piece and a tail. A plasma membrane envelops the whole body of sperm. The sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by cap-like structure acrosome. The middle piece possesses numerous mitochondria, which produce energy for the movement of tail that facilitates sperm motility essential for fertilization. Middle piece is called powerhouse of sperm. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% sperms must have normal shape and size, and at least 40% of the 60% must show vigorous motility. Sperms released from the seminiferous tubules are transported by the accessory ducts. Secretions of epididymis, vas deferens, seminal vesicle, and the prostate glands are essential for the maturation and motility of the sperms. The seminal plasma, along with the sperms, constitutes semen. The functions of male sex accessory ducts and glands are maintained by the testicular hormone called as androgen. Nebenchern is a part of sperm. 2. Oogenesis. The process of formation of mature female gamete is called as oogenesis, which is marked by differently from spermatogenesis. Oogenesis is initiated during the embryonic development, where a couple of million gamete mother cells, that is oogonia, are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into prophase 1 of the meiotic division and get temporarily arrested at that stage called as primary oocyte. Each primary oocyte then gets surrounded by a layer of granulosa cells and then called as primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layers of granulosa cells and a new theca called a secondary follicle. Secondary follicle soon transforms into a tertiary follicle, which is characterized by a fluid-filled cavity called as antrum. The theca layer is organized into an inner theca interna and an outer theca externa. It's important to draw your attention that it is at this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. It is an unequal division resulting in the formation of a large haploid secondary oocyte and a tiny first polar body which eventually degenerates. The secondary oocyte retains bulk of the nutrient-rich cytoplasm of the primary oocyte. First polar body born out of the first meiotic division divide further or degenerate. Actually, it degenerates, and we are very certain about this right now because it degenerate and it gets mixed with the body wall. The tertiary follicle further changes into the mature follicle or Graafian follicle. So mature follicle is also called as Graafian follicle. The second oocyte forms a new membrane called as zona pellucida surrounding it. The Graafian follicle now ruptures to release the haploid secondary oocyte, that is ovum, from the ovary by the process called as ovulation. Extrusion of secondary polar body from egg nucleus occurs after the entry of sperm but before fertilization. Mature Graafian follicle is generally present in ovary of healthy human female around 11 to 17 days of menstrual cycle, because the menstrual cycle or the ovulation exactly is on 14th. So an average day is 11 to 17. The outermost cover of the ovum at ovulation is called as corona radiata. Menstrual cycle. The reproductive cycle in female primates, example monkeys, apes and human beings, is called as menstrual cycle. The first menstruation begins at puberty and is called as menarche. In human females, menstruation is repeated at an average interval of 28 to 29 days. The cycle of events starting from one menstruation till the next one is called as menstrual cycle. One ovum is released, that is ovulation, during the middle of each menstrual cycle, that is on 14th day approximately. Menstrual cycle is divided into three phases. One is menstrual or bleeding phase, where menses occur. The cycle starts with the menstrual phase when menstrual flow occurs and it lasts for 3 to 5 days. The menstrual flow results due to breakdown of endometrial lining of the uterus and its blood vessels, which forms liquid that comes out through the vagina. Menstruation only occurs if the released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy. However, it may also be caused due to some other underlying causes like stress, poor health, tension, etc. Follicular or proliferative phase. The menstrual phase is followed by the follicular phase. During this phase, the primary follicles in the ovary grow to become a fully mature Graafian follicle, and simultaneously the endometrium of uterus degenerates through proliferation. These changes in the ovary and the uterus are induced by changes in the levels of pituitary and ovarian hormones. The secretion of gonadotropins LH and FSH increases gradually during the follicular phase and stimulates follicular development as well as secretion of estrogens by the growing follicles. Both LH and FSH attain a peak level in the middle of the cycle about 14th day. Rapid secretion of LH leading to its maximum level during the mid-cycle, called as LH surge, induces a rupture of Graafian follicle and thereby the release of ovum, which is called as ovulation. During evolution, the concentration of LH, FSH and estrogen is high. During ovulation, the concentration of LH, FSH and estrogen is high. Luteal or secretory phase, that's the third phase. The ovulation, that is ovulatory phase, is followed by the luteal phase, during which the remaining parts of the Graafian follicle transform as the corpus luteum. Corpus luteum is a temporary endocrine gland in the human body. The corpus luteum secretes a large amount of progesterone, which is essential for maintenance of the endometrium. Such as endometrium is necessary for implantation of the fertilized ovum and other events of the pregnancy. During pregnancy, all events of the menstrual cycle stop and there is no menstruation. In the absence of fertilization, the corpus luteum degenerates. This causes disintegration of the endometrium, leading to menstruation, marking a new cycle. No new follicles develop in the luteal phase of menstrual cycle because both LH and FSH levels are low in the luteal phase. In human beings, menstrual cycle ceases around 50 years of age, that is termed as menopause. Cyclic menstruation is an indicator of normal reproductive phase and extends between menarche and menopause. changes in GnRH, that is gonadotropin releasing hormone pulse frequency in females is controlled by circulating levels of estrogen and progesterone. Progesterone level is high during the past ovulatory phase of menstrual cycle. Next is topic number 7, fertilization and implantation. During copulation or coitus, semen is released by the penis into the vagina, called as insemination. The motile sperm swim rapidly, pass through the cervix, enter into the uterus, and finally reach the ampullary region of the fallopian tube. The ovum released by the ovary is also transported into ampullary region where fertilization takes place. Fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. This is the reason why not all copulation lead to fertilization and pregnancy. The process of fusion of a sperm with an ovum is called as fertilization. Capacitation refers to changes in the sperm before fertilization. Capacitation occurs in female reproductive tract. Capacitation occurs to the sperm before fertilization, which are the changes. During fertilization, a sperm comes in contact with the zona pellucida layer of the ovum and induces changes in the membrane of zona pellucida that blocks the entry of additional sperms. Thus, it ensures that only one sperm can fertilize an ovum and avoids polyploidy. Zona pellucida layer of an antral follicle is acellular. Note the point. Zona pellucida layer of an antral follicle is acellular. In human females, meiosis II is not completed until penetration of sperms, that is fertilization. The secretion of the acrosome helps the sperm enter into the cytoplasm of the ovum through the pellucida and the plasma membrane. This induces the completion of the meiotic division of the secondary oocyte. The secondary meiotic division is also unequal and results in the formation of second polar body and a haploid ovum or oocyte. Soon, the haploid nucleus of the sperms and that of ovum fuse together to form a diploid zygote. Acrosomal reaction of the sperm occurs due to its contact with zona pellucida of ova. 50% of sperms carry the X chromosome, while other 50% carry the Y chromosome. After fusion of male and female gametes in the zygote would carry either XX or XY depending on whether the sperm carry X or Y fertilized the ovum. Sex of the baby is determined by the father and not by the mother. The mitotic division starts as zygote moves through the isthmus of the oviduct called cleavage towards the uterus and forms 2, 4, 8, 6 daughter cells called blastomeres. The embryo with 8 to 16 blastomeres is called as morula. The morula continues to divide and transforms into blastocyst as it moves further into the uterus. The blastomeres in the blastocyst are arranged into an outer layer called as trophoblast and an inner group of cells attached to trophoblast called as inner cell mass. Morula is a developmental stage between the zygote and blastocyst. The trophoblast layer then gets attached to the endometrium and inner cell mass gets differentiated as the embryo. After attachment, the uterine cells divide rapidly and covers the blastocyst. As a result, the blastocyst becomes embedded in the endometrium of the uterus. This is called as implantation and it leads to pregnancy. In the human female, implantation occurs almost 7 days after fertilization. Topic: Pregnancy and embryonic development. After implantation, finger-like projections appear on the trophoblast called as chorionic villi, which are surrounded by the uterine tissue and maternal blood. The amnion of mammalian embryo is derived from ectoderm and mesoderm. The amnion of the mammalian embryo is derived from the ectoderm and the mesoderm. The chorionic villi and the uterine tissue become interdigitated with each other and jointly form a structural and functional unit between developing embryo, fetus and maternal body called placenta. The placenta facilitates the supply of oxygen and nutrients to the embryo and also removal of carbon dioxide and excretory waste materials produced by the embryo. The placenta is connected to the embryo through an umbilical cord which helps in the transport of substances to and from the embryo. Placenta also acts as endocrine tissue and produces several hormones like human chorionic gonadotropin (HCG), human placental lactogen (HPL), estrogens and progesterones. In the later phase of pregnancy, a hormone called as relaxin is also secreted by the ovary. During pregnancy, the levels of other hormones like estrogen, progesterone, cortisol, prolactin, thyroxine, etc. are increased several folds in the maternal blood. Increased production of these hormones is essential for supporting the fetal growth, metabolic changes in the mother, and maintenance of pregnancy. Immediately after implantation, the inner cell mass, that is embryo, differentiate into an outer layer called as ectoderm and an inner layer called as endoderm. A mesoderm soon appears between these two layers. It needs to be mentioned here that the inner cell mass contains certain cells called as stem cells which have the potency to give rise to all the tissues and organs. During pregnancy, degeneration of corpus luteum is prevented by HCG, human chorionic gonadotropin hormone. During pregnancy, degeneration of corpus luteum is prevented by HCG. In normally pregnant women, HCG activity shows high level of circulating HCG to stimulate the synthesis of estrogen and progesterone. Embryo. In human beings, after 1 month of pregnancy, embryo's heart is formed. The first sign of growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. By the end of the 2nd month of pregnancy, the fetus develops limbs and digits. By the end of 12 weeks, that is first trimester, most of the major organ systems are formed. For example, the limbs and external genital organs were well developed. The first movements of the fetus and appearance of hair on the head are usually observed during the 5th month. By the end of about 24 weeks, that is 2nd trimester end, that is almost 6 months are end, the body is covered with fine hair. Eyelids separate, eyelashes are formed. By the end of 9 months of pregnancy, the fetus is fully developed and is ready for delivery. Parturition and lactation. The average duration of human pregnancy is about 9 months, which is called as gestation period. Vigorous contraction of the uterus at the end of pregnancy causes expulsion or delivery of the fetus. This process of delivery of the fetus, childbirth, is called as parturition. Parturition is induced by a complex neuroendocrine mechanism. The signals for parturition originate from the fully developed fetus and the placenta, which induce mild uterine contractions called as fetal ejection reflex. This triggers the release of oxytocin from the maternal pituitary. Oxytocin acts on the uterine muscle and causes stronger uterine contraction, which in turn stimulates further secretion of oxytocin. The stimulatory reflex between the uterine contraction and the oxytocin secretion continues, resulting in stronger and stronger contractions. This leads to the expulsion of the baby out of the uterus through the birth canal, called as parturition. Soon after the infant is delivered, the placenta is also expelled out of the uterus, that is afterlife. The important components of initiation of parturition in humans are release of oxytocin, synthesis of prostaglandins, increase in estrogen and progesterone ratio. These are the important components of initiation of parturition in humans. In mammary gland of the female undergo differentiation during pregnancy, starts producing milk towards the end of pregnancy by the process called as lactation. The milk produced during the initial few days of lactation is called as colostrum, which contains several antibodies absolutely essential to develop resistance for the newborn babies. Breastfeeding during the initial period of infant growth is recommended by doctors for bringing up a healthy baby. The correct sequence of human reproduction is gametogenesis, gamete transfer, syngamy, zygote, cell division or cleavage, organogenesis, and cell differentiation. So that was the whole chapter. Hope you guys enjoyed it. I tried to cover the whole points of the NCERT textbooks. If you like the video, please do subscribe the channel and like the video. Don't forget to hype it. So thanks for watching and do check out our channels for more updates. Thank you.